Hello, I'm Samantha Beasley. I'm in the Pharmaceutical Sciences PhD program. I'm currently a fourth-year grad student. I came in through the Medicinal Chemistry and Pharmacology program, and I chose it because it actually gave me the opportunity to try everything out for about a year. So I got to try chemistry, biology, and pharmaceutical sciences, as well as pharmacology, and basically figure out where I felt most comfortable, and I ended up choosing pharmaceutical sciences. With the MCP program, what I really liked about it is that I had quarter-long rotations, so I really got to get a feel for the lab, and I've heard other rotations are only 3 weeks long or less, and you don't really get to do any hands-on work. And with this program, I was running experiments. I was actually doing my own personal projects for quarter-long rotations, so I actually got to try out the research before I dedicated my life to it, because you know, grad school is like signing your life for 5 years, so definitely want to make sure you like it. So the Spitale Lab focuses on RNA. We study it in both terms of chemistry and biology. And you may be thinking, what is RNA? And you might remember that DNA is transcribed into RNA, which is then translated into protein. And that was like the long thought that RNA is just this middleman shuttling from the genetic information of DNA to make some proteins. Turns out that RNA can actually do a lot more functions. It can actually interact directly with DNA and turn genes off or on. It can interact with proteins and shuttle them to wherever they need to go inside of the cell, or it can even act on other RNAs. So our lab really wants to come up with these chemical tools as well as biological tools to study all these RNAs, because there's so many out there now, and there's so many new functions that we still have yet to learn. So to give you a few examples of what we do research-wise in the Spitale Lab, we are trying to focus on how to get orthogonal nucleosides into RNA. And so one of the first things that I set out to do was to synthesize some orthogonal nucleosides that have azide handles on it. And from this, we could take these nucleosides, put them into cell culture, allow them to incubate for over a certain amount of time, and then extract all of the RNA out. So now you're going to have your RNA full of these little azides, and through copper chemistry we can then click them out, and we can take all of that RNA out and see if these even made it into nascent RNA inside of the cells. So we can prove that through dot plots, where all of these streaky black marks that don't look very pretty are actually RNA incorporation, with number 3 being the one that was incorporated the best. So now that we knew we could make orthogonal nucleosides that were capable of being incorporated to RNA, the next idea is that we wanted to put some sort of cage on our nucleosides that would then only be removed in the presence of specific enzymes. So as you can see here, we would, same idea of giving our probe to cells, but now we're relying on an enzyme that's present to then activate our probe to then be incorporated into an RNA. And the reason why we would want to do this is because we can now do cell-specific RNA labeling by making this enzyme be expressed inside of our cell line of interest. And then a few examples of these specific enzyme pairs was the UPRT enzyme, where we found out we can make these uracil analogs, and it will then be incorporated into a monophosphate nucleotide, which can then be incorporated into RNA. We found that our 5-ethynyl uracil analog worked very well. And then additionally, we found another enzyme pair of the penicillin G amidase enzyme, which will actually selectively cleave off this group. So we attached it to our 2'-azidoadenosine probe we had already made previously. And now we have a new enzyme pair for doing cell-specific RNA labeling. And then so from our enzyme and nucleotide pairs, we then did some cell imaging, which is pretty cool. So you can see that in panel A is our UPRT cells and non-UPRT cells, and all of the green signal is the RNA that or the nucleotide was then incorporated into RNA. So yes, it is all of our RNA. And then in panel B is our PGA cell lines and no PGA cell lines, and you can see that all of the red is now all of that RNA signal from our nucleotide that was caged. And then the really cool thing in the end was that our biologist Kim, who's awesome, mixed both UPRT and PGA cells together, so you can see that the green labeling is only in the UPRT cells and the red labeling is only in the PGA cells, which is pretty awesome. So we achieved what we set out to do, is to get cell-specific RNA labeling.